Hi students. So today we are continuing again with the identification of mutants. We had mentioned about different mutants, and one among them is a biochemical mutant. Hope so you remember this. A biochemical mutant is a mutant which would undergo some mutation, and then it will lose the ability to synthesize a particular essential molecule such as an amino acid or a nucleotide. So, as a result, if you grow them in a particular type of media which lacks that particular essential nutrient, they will not be able to grow in that particular media. So, the mutants which are unable to grow on a media lacking that molecule, but grows when the molecule is provided, is referred to as an auxotropic mutant. And they are said to be auxotropic for a molecule they cannot synthesize. Now, the example that we have is sometimes you have an organism, okay, and that organism is undergoing a UV mutation, and after that it is not able to synthesize tryptophan. So, it will not be able to grow in a media in which tryptophan is not there, because if tryptophan is an essential amino acid for it, probably in the media where tryptophan is not there, the organism will not be able to grow, and that organism and that mutant is referred to as an auxotropic mutant. And the wild strain from which the mutant arose is a chemoorganotrop, and which can grow on the minimal media containing only salts, that is only salts provided nitrogen, phosphorus and carbon all is there, but this essential amino acid is also not there. The wild strain which can grow even in the absence of that particular tryptophan deficiency is referred to as a prototrop mutant. Then another type of biochemical mutant is a resistance mutant. Sometimes what happens after the mutation, and wild type strain will become resistant to some antibiotic or anything like that, or it can be vice versa also. So, resistant mutants are those mutants which can be identified based upon their resistance to certain antibiotics. So, let us go into the techniques which we can identify these. Such mutants, biochemical mutants, can be detected by using either a negative or a positive selecting technique. Now, what is a negative selecting technique? A negative selecting technique is a one which we use in the case of auxotropic mutants. That is, you will grow them in a media missing a particular nutrient that the mutated bacteria cannot synthesize. So, the bacteria cannot synthesize the nutrient, will die. Now, coming to the next one is the positive selection technique. In the positive selection media, which contains a specific substance called the antibiotic here, to determine those resistant mutants. Sorry, I got interrupted in between. So, in the case of biochemical mutants, you can identify them either using a negative selection technique or by a positive selection technique. So, let us go into the details of that. Now, in the case of positive selection of mutants, you will grow them in a media. Now, this happens in a case where you are trying to get a recombinant strain. Now, for example, you have a when you take organism and it is undergoing some mutation, okay, and sometimes what happens, it will become resistant to some antibiotic, and some of them, the wild strains, will not be resistant to some antibiotic. So, in such case, to identify the resistant as well as a non-resistant organisms, or non-resistants are referred to as a sensitive organisms, you will plate them onto two different media, the one which is containing an antibiotic and the other one which is not containing an antibiotic. So, like that you can differentiate whether they are being resistant or whether they are being found to be sensitive. So, the medium with penicillin refers to the organisms which are being found to be resistant, and the medium without penicillin, after plating them, say all the organisms have been present in a single tube. After that you take a required amount, now 100 microliters of that you spread plate onto this, 100 microliters of that you spread plate onto this, and the organisms which grow in the medium with penicillin indicate that they are being found to be resistant to penicillin, and the organisms which will grow in the medium without penicillin refers to those cells which are being found to be sensitive. Now comes to the next one technique. You can also use another technique called the replica plating technique. Okay, this is a technique which is mainly used to identify the auxotropes. So after growing the culture in the presence of a mutagen, first you grow it in a media. Okay, this is referred to as a master plate. You will grow it in a media which is been containing that particular amino acid. Okay, you remember what is a auxotropic mutant. So you grow it in a complete media which already contains the amino acids. So in this plate, what happens, both the prototropes as well as the auxotropes will grow because of the presence of that particular amino acid. After that, you grow it into you prepare a replica of that particular plate onto two media. One media will be the same complete media, and the next one is a media minus lysine. It can be any amino acid here. Here in this picture they have depicted lysine. So what you do, you take a velvet surface one, which is velvet surface is been attached to some, you can have different things. You can either have a Petri plate itself or a conical flask at the bottom of a conical flask, whatever it is, according to your convenience, something which will fit into the Petri plate. It should be something which will fit into the Petri plate. On one end you will cover that with a sterile velvet cloth, and you will touch with that velvet surface will be touching on the master plate, and then it will be replica plated. With that imprint you will prepare one on this as well as on this plate, and you will incubate both the plates. And this is, since it is a complete media which already contains lysine, what happens, all the strains will grow over here. But in the next plate, what happens, lysine is not there. So only those microbes which can synthesize lysine will grow here. And so there would not be growth of the lysine auxotrope. So you will compare. So when you look here, there are some strains which are not being present here, but it has been present in this. Like that you will compare and you can identify the lysine auxotrope. And here the culture lysine auxotrope LS minus LYS minus strain will be grown in the media with lysine. It will be grown here, but it will not be grown in this media. So you can identify, of course, this is a lysine auxotrope. Now same technique can be used for different amino acids, like, or you can use two or three amino acids also can be used together to identify the auxotropic mutants. And this is the replica technique shown in a different here. This is a tetracycline resistance which has been shown here, and all the colonies will grow here, and this is a tetracycline mutant. As only the mutants can grow here. Okay. It depends upon, it is a replica technique using for the identification of antibiotic resistance also you can do it here. For that technique also you can use this replica technique, that is, this is a media which is containing tetracycline. So only those media, only those organisms which can be tetracycline resistant will grow here, but in a normal media all the organisms will grow, but in this media only those organisms which are tetracycline resistant will grow here. Like this we can use the replica technique for the identification of both biochemical mutants as well as the auxotropic mutants. Hope so this is clear to you, and with this we come to an end to the identification of mutants by, especially the microbial mutants, by various techniques. Thank you.